So we already talked about ribosomal RNA, and ribosomal RNA is RNA that is not going to be translated into proteins, but it's going to have a specific function and role inside our cells. In our case, it's going to compose a part of the ribosomes, which is going to help us create and translate different mRNA into proteins. We also have something called tRNA. tRNA stands for transfer RNA. And the tRNA is going to help us transfer different amino acids from the surroundings of the cell to the ribosome in order to help us create a sequence of amino acids, therefore the proteins. So tRNA, once the RNA of the tRNA is transcribed from the DNA, will start as a single-stranded RNA, but because of the intermolecular forces that are going to be able to cause inside and between the RNA molecule, it is going to fold into a specific shape, and this specific shape is something that resembles a clover leaf. Clover leaf. And every time we have a tRNA molecule, you will see that there is a very unique and specific shape that this tRNA is going to fold into. This tRNA molecule, and let me give you a better zoom in into the tRNA molecule, is going to have very specific and important places. It's going to have the acceptor arm, which is going to accept through specific chemical covalent bonds the amino acid. It is going to have different, some sort of hairpin-like formations because of the hydrogen bonds and intermolecular forces you can form between the parts, different nucleotides when it comes to this RNA. And it will also have, which is the most important part we need to study, the anticodon that is going to be the exactly complementary codon that is going to fit one of the codons we already studied on. So tRNA will be able to carry a specific amino acid. This specific amino acid will comply to the codon sheet that we saw, the codon chart that we saw a few lessons ago. And therefore, you will see and you will know exactly which amino acid is going to fit our codon. For example, if we look at the codon chart, you will see that AUG, which is our chart codon, is going to give us methionine. Methionine is usually the first amino acid in the sequence, and it's the AUG codon result. Now, if we have AUG, which is 5 to 3 in our messenger RNA, the anticodon will be 3 to 5. And it is going to be the exact complementary RNA for this codon. So AUG will give us uracil, adenine, and cytosine. And you will see that when we have the anticodon of UAC from 3 to 5, the codon will be AUG. And if you have you will have here UAC. This amino acid that is going to be chemically covalently connected to the tRNA is going to be methionine. And it complies to all of the different tRNAs. So each one will give us a specific amino acid, and the anticodon will fit the exact codon that we already talked about from the chart. You will have multiple possibilities for the codon as we talked about in the previous video. In the previous videos we were talking about that we have multiple possibilities for different codons to have one single amino acid. And it's caused from the fact that this third place can form something funny called a Wobbly Connection, which it's from the Pearson book. I just quote in the book. And basically it's you don't fit it all the way, but it still fits. So you can have different nucleotides when it comes to the third position, the third nucleotide, but you will still have a strong enough connection to create and to give the same amino acid. So you saw if you remember from the codon chart, that the third nucleotide changes between different codons, but it will still give us the same amino acid. This is why, and this is where it comes to actually to life. You will have the same codons, the same nucleotides, in the first and the second position, but the third one can slightly change between different amino acids, but it will still fit some amino acids, some tRNA, and it will still give us the same amino acid. And the process itself is pretty interesting. You will see that we have the tRNA coming inside the ribosome one after one after one, and then you will have the catalyzation of the peptide bond formation between the different amino acids, one after one after one. So one tRNA will join, will stay inside the ribosome, then the other will join, they will form connections, then it will just leave. Then another one will join, and then another one will join, there will be two inside, they will form peptide bond together, and then it will leave. Therefore. It's very interesting to see how it happens, and we are going to study it in future videos. But for now, just understand that, let's summarize this video, that one tRNA molecule is an RNA molecule that is going to bring us on one side the amino acid, and on the other side it's going to have an anticodon sequence that is going to be complementary to the codon of the mRNA that is being and waiting for us to be translated inside the ribosome. The tRNA will be inserted, will enter the ribosome one after one, and the different tRNAs will be able to stay together and close to each other, and the overall structure, the ribosome and the different enzymes that we are going to study are going to help us form peptide bond between the amino acids. Therefore, we will have the elongation of a protein, that is of a polypeptide that is eventually is going to be called a protein. And that's it. I hope this video made sense. In the next video, we are going to elaborate on the translation process, and we are going to see how it actually happens.